Hello, Miss Rasmika, can you hear me? Miss Rasmika, I don't put Rasmika, you cannot hear you yet. Thank you.
SIR laboratory under Chemical Engineering and Process Development Division. Her main work involves cheminformatics analysis of mass spectrometry and cheminformatics investigation of chemical defense and plants. And she also has a work experience as junior research fellow at CSIR laboratory, Chemical Engineering and Process Development Division, and also has worked as a patent analyst. And to her credit, she has excellent academic records. Like she has been awarded with Senior Research Fellowship for PhD Life Science CSIR UGC NET exam. She has published her research articles in several high impact journals. And currently she is working as a senior research scientist at Syngenta, United Kingdom. As an AI researcher in drug discovery, over 7 years of experience in biological sciences like cheminformatics, computational biology, and biotechnology. Now, with great privilege, I humbly invite Ms. Divya Karade to take over. Ma'am, please.
Yeah, thank you very much for this very big introduction. I really highly obliged for that you researched so much about me. I never expected this. Anyways, thank you very much. So yes, currently I am working as a senior research scientist in Syngenta Gillett's Hill International Research Center, which is a part of Syngenta in UK. So Syngenta is a multinational company, so one company is in UK also. So you have already explained my journey, but I want to explain to the students to just inspire them to make them India proud. So in between of a PhD, during PhD, I did some other work. Means my PhD work is related to chemoinformatics designing of drugs, drug molecules, drugs like medicines. For the students who don't know what drugs is, it's not cocaine and everything. These are the pharmaceutical drugs. In science, we call them drugs. Drugs and crop protection agrochemicals like pesticides, insecticides. That's what my PhD was about. But in between that, during the COVID lockdown, when everywhere lockdown was going on and people are scared about coronaviruses. So that time, our Indian government has initiated several programs, and many things were stopped in between that. My PhD research work has been stopped. It's completed, but the process was not going on ahead because of the COVID lockdown. We can't meet each other. So so that time I started learning myself regarding the artificial intelligence, machine learning and deep learning technologies using whatever study materials are present in the internet. So I just studied all those things in the COVID lockdown, in the lockdown period of, you can say, 2 years. So from there, I got that much importance, that much value. The work gets so much valued. So that's why I'm here right now. I'm working here as a senior research scientist. So my goal to inspire all the peoples who think that they can't do anything, they can't move ahead. So the thing is, if they will try, they can do anything. Means they just need to practice data science. If they want to go to data science field, they just want to do data science. To learn data science is to do data science. So if they learn it, because everything is present, all study materials. Internet has a huge kind of study materials, all kind of videos, study tutorial videos. So I just want them, if you really want to go into the data science field, then please go ahead and learn and see and explore what material is there in our Google or in internet. So from there you can learn many things. So this is one of the biggest technology which empowers us while we are working at we are in home also, means we can do many things by working from home only also. So this was the biggest impact which I had experienced in my life, and I want that everyone should try this, everyone should do this, and make India proud. So this was my journey in between that PhD, which valued a lot. So so today I have to share my screen. Share. So is that visible to everyone? Hello? Is my screen is visible to everyone?
No, ma'am. Again, can you please share again?
Screen. Now?
Yes,
is that
now it is.
visible?
Yes, ma'am, it is visible.
Okay. So thank you very much for inviting me for this webinar. And as we all know, the topic of this webinar is the development of web application for drug discovery. So so this is not the topic which I have done. This is not the work which I have done during my PhD. This is some extra work which I did during the COVID lockdown, and which moves very fastly and get very popularized so much. So it gives me some national and international awards also because of that. So how I did this? So first, before that, we need to know that what is the basic goal, what is the basic science behind this. So as I already spoke that I built this web application, AI drug app, which is known as AI drug app, because of the coronavirus, to screen the molecules which can be drug molecules, which can build as drug molecules, and we can screen those molecules through this web application. So there are so many other things I have included in this web application. So as we all know, many people were died, and everywhere people were died. There was no cure, no medicines. So Indian government initiated some drug discovery hackathon programs in the India International Science Festival programs, from which they are inspiring, they are encouraging people to do something, to design, so that we can discover some new molecule, new drug-like molecule towards this coronavirus, to fight this coronavirus. So so the drug discovery is a very long process, and it's a very costly process. It requires billions of dollars of investments, and it requires decades of years of research. And after this also, what we get, the result output of the drugs is very low, means only few, less than 12% of drug molecules gets approved by the FDA. So that's why we need an efficient treatment. We need proper therapeutic targets and drug-like compounds. And how we can do that fastly? We have to use some tools. We have to use some softwares which can screen those molecules from a very large databases, which are already present in various databases like Campbell, drug. There are so many related databases where so many molecules are already present. And from them, from these databases, it is very difficult to which molecule, which particular molecule will act as a drug molecule for a particular disease like corona or anti-diabetic or anti-cancers. So we need a tool, we need a software, and here AI, artificial intelligence come into the picture, because it solves the problem. AI, everybody are using AI. AI is a part of data science. So okay, so AI for drug discovery and development, the market for this is growing very increasingly. It's growing exponentially, and the people are putting their money, putting all their investments in AI related things. So one of the part is in drug discovery. So data science is not only for drug discovery, but we can use it for various other purposes, like for agrochemical discovery, for engineering also. AI are using engineering part for medical field, for robotic surgery, like that. AI has immense application. So today I'm going to speak about drug discovery part, how we can apply AI in drug discovery. So as I said, there are a large number of databases and containing large number of molecules, but we don't know which molecule, which particular molecule will act as a agent, act as a medicine or treatment for that particular disease. So we need to find that, and this is called virtual screening. Virtual screening is to screen those molecules from the big lake of database. So so many people are many kind of people are involving for this process, like chemists, biologists, drug discovery enthusiasts, and so many informatics people are involving in this. There's a big group of scientists here who are working on the screening of these kinds of molecules from big databases. So how we can do it fast? So so the basic concept is as we all learned during our when we were in school, we learned that lock and key system of a protein and molecule, enzyme and molecule, you must have remembered this. So when the enzyme is a kind of protein, so when the molecule is attaches to that enzyme, to that protein, then it activates that enzyme. When it can't binds, it is unable to bind to that enzyme or protein, then it enables to it it doesn't activate, it doesn't show anything to that. So if the molecule binds to that enzyme, then it activates and it will create some chemical reaction, some biochemical reaction in the body. So this is the basic principle. So what kind of biochemical reactions these molecules can have after binding various kinds of proteins, various kinds of enzymes which are already present in our body, which are related to a particular diseases. So this is the basic principle behind all these drug designing thing. So this is the same thing what we need to find out. We need to screen those molecules which fits exactly according to the shape and size to the enzymes, to the protein molecules. So we need to screen those molecules. So one thing comes, that QSAR and drug design. QSAR is a quantitative structure activity relationship. It means that what structure of molecule or what structure of enzymes or proteins will fit with each other correctly, so that it will create some activity, whether the molecule is inactive or active for that particular enzyme which is related to a particular disease. So this thing we can we calculate it in some kind of scores or in some kind of equation? Can we do that? So this is called QSAR, the quantitative structure and activity relationship. So we find the relationship between the structure and activity, the structure of the molecule with that protein, and what is the activity. So whether it is active or inactive towards that protein or not. So this, all the equations is built, and the process is known as a quantitative structure activity relationship, QSAR, and from which we can find out we can predict the new molecule. Means there is already, okay, the next slide. So there are so many databases, as I've already told, so many databases are there related to compounds that for example, Kimball database. So there are so many molecules. If you'll go to that database, there are so many molecules present which are linked to some particular proteins, and those proteins must have some of their significance for particular diseases. So these collections are already there, which are extracted from the literature, from experimental, like the experiments which are going on, or which will become in mice or rats. So these kinds of experimental databases are already present through literature and in databases. So these molecules and these compounds were used in these QSAR modeling technique. So these where we identify the equation of structure activity, means which structure will fit to that particular protein, to a new protein, for example, or to a new molecule. So it makes a kind of equation, and it uses so many softwares and tools. So it has been done actually from very previously we have done that, but there were not particular softwares which runs the big data in a few minutes, which can give the very accurate results. Means for quasar modeling, there are already so many kinds of tools and softwares are available, but they are not that fast, and not that they are not giving so much accurate result for screening those large databases, to screening those molecules from those large databases. So that's why we have nowadays people are including artificial intelligence like deep learning or machine learning, traditional machine learning, to these quasar modeling technique to synthesize those new compounds. After screening those new molecules towards a particular disease, they synthesize chemically these new compounds. So as I said, there is a large amount of data already present, years of experimental data, years of researches, they have been collected in various databases, various publications, various patents, and so many chemical databases and libraries are present. And one need to explore that data, one need to, yes.
Sorry for the interruption. Can you share your screen, ma'am? We can't able
Isn't
to.
it visible?
No, ma'am, no.
Okay, I'm just sharing my screen. Okay, please tell me when it is not visible. Is it visible now?
Yes, ma'am, yes.
Okay.
Thank you, ma'am. We can
Okay,
continue.
from when? Yeah, from when it was not visible? Have you saw this?
No, ma'am, from the long time it was not able to.
Oh, okay. Have you seen this from when? From when? Have you seen this this picture, this slide? Hello.
Yes, ma'am. Yes, ma'am.
You have seen this, okay? And this
Yeah,
one?
yes, ma'am.
Okay, and this?
Yes, ma'am.
Okay. Okay, so this is the slide now, which you must not able to see. So I'm talking about the big data. Is it visible now?
Yes, ma'am. Yes, ma'am. You can continue.
Okay. Okay.
Thank
So there are so many, yeah. So there are so many ongoing researches, which means researches are continuing, means it's already going on, and it produces so many large amount of data. But one needs to manage those data. So there's where the chemoinformatician, the computational biologist, they come in this thing to analyze this data, to manage this data. So we need those people, and there are very small number of people right now in the world. So we need more people to manage all these big data, because the data is so large it is spread in all over the areas related to drug designing, to agrochemicals, to robotic surgery, to many other things. It is it is a vast amount of data. There are vast amount of data is present, and we need more people to manage this data. So the chemoinformatician, computational biologist, bioinformatician, these are the people who can help us to analyze this data. So so that's why AI is also one of the thing where we can do where we can use these data in a short amount of time. We can analyze this data very accurately to give accurate predictions. So this is the AI. So AI is basically how we can connect the machine intelligence and the human intelligence. How we can connect it means how we can program software or tool or robot or anything which will think like human. Means it is not just like yes or no. We need more data. We need more analyzation of the data. So that's what AI do. So this is a branch of computer science, but it is nowadays is applicable everywhere. And I want to emphasize the fact again that if one needs to go ahead in their career, or we need more if we need more discoveries, then one should go through the AI thing. Means it is must have. Means for every field we need to learn AI. The little means it's not of course it's a field of computer science, but nowadays in all areas they are giving such kind of course. Means in universities people are adding these kinds of machine learning and AI courses, and which are involved in various disciplines like biological science, chemical science. So it's a need of the future, for the future. So that's why so this is what about AI. So these are some applications of AI in drug discovery, how we can use artificial intelligence for drug property prediction, for designing novel molecules which can be a new drug for a unknown disease or a non-disease, any kind of diseases, and to predict what will be the interaction, how the drug will behave when it binds to the target like RNA, DNA, any sequential DNA, or any enzymatic, any protein target. So these kinds of studies we can in these kinds of studies we can use artificial intelligence. So data science is a very large field, and it comprises deep learning, machine learning, artificial intelligence, and deep learning and machine learning are the part of artificial intelligence, and artificial intelligence is a whole. And data science is a part of artificial intelligence, because artificial intelligence is very big arena, but we are in some disciplines we are using only some part of that artificial intelligence. We are not using everything, because there is so much, and so much our researchers are still going on. It's still increasing. So many people are discovering so many codes and tools related to artificial intelligence. It's still going on. So we don't know where it will stop. And yes, maths, statistics, visualization, exploratory data analysis are the part of data science, and it is very much required for the machine learning part to confirm the result, confirm the output of machine learning part. So these are the things. So from where you can learn all these things, all these data science related things. If you are from biology background like me, I didn't know before my PhD that is there anything like, or I need to do anything like coding or programming like that. So where we can start, So first thing, YouTube. YouTube is a very good channel, good source of learning, because everything is free in there. All kind of tutorials you can find if you want to do, like for example, Data Professor and Simply Learn. They provide so many good tutorials regarding web applications, how to develop web applications, how to code for web applications, and they start from very basics. So YouTube has a very large number of tutorials present in there in the form of videos. Second is LinkedIn Learning. Okay, it will cost some amount from you, but it has also some good courses like LinkedIn Learning, Coursera. These kinds of sites are there, and you can learn from research papers which are freely available from PubMed or Google Scholar kinds of databases, and online courses, as I already said, Coursera. And there are so many blogs people do write who are related, who are working in this area in analytics with Vidya, Medium, and towards data sciences. So you can get all these study material, or if you want to start something, then you can start from here, start visiting, or you can log in or sign up or be a member of these sites. You will get notification in your Gmail account or in your mail whatever mailing system you are using. You will get all those new data, all those new kind of researches, blogs, everything, tutorials. You will get in your, the notification you will get in your mail, and you can get access to that. So these are some of them are very freely available, which one can learn, start learning. This is the basic thing, that how deep learning or machine learning works for predicting the new molecule. So is that visible now? Is this slide is visible now?
Yes, ma'am. Yes.
Hello? Yes.
Yes, ma'am.
Okay. So there is some process in machine learning or deep learning, that how one can predict the data or molecule, for example. So there are some procedure. So first we need to identify a problem. What problem one needs to solve using machine learning. So, and to prepare that data. So as example I showed before in previous slides, there are large amount of databases are present, and so many data are already available in literature and everything. So we need to collect that data. Okay, so this, and prepare that kind of data in, for example, in Excel sheet, so that it can be a input file for that particular uh, process of prediction. Then we need to select the features. Okay, what are features? So for example, a molecule has various features like melting point, its a molecular weight. So these are also called descriptors. So as I said about QSAR techniques or QSPR analysis, so there are various kinds of descriptors we can generate. These kinds of properties, descriptors are nothing but the properties of the molecules. So there are various other tools are present. These are called QSAR tools for generating descriptors, are already present, and some are very freely available, like Paddle software is available, and RDKit is also available, which is free. From that you can easily generate those descriptors, those properties for your kind of molecules, for your set of molecules. So from that you need to generate those features or descriptors or properties. It is the same thing, and using those from that you the machine will the code will automatically select those features which are important, which are valuable for the kind of activity we want to predict. For example, the endpoint we want to predict is whether the molecule is active or inactive towards a particular molecule. So on the basis of that, it will select some features. It will, after selecting the feature, it will split the data into training and test set. Then after splitting the data, it will build the AI model, the machine learning model. It will train the model based on the training data, and train the model means it will connect the activities of the molecules, like active or inactive of the molecules, towards the descriptors, towards these properties. So this is called training. And in practically, we need to train so much time, means it takes a time and the skill to train that model, to make it fit, so that we can use that model to evaluate the new molecule, to evaluate the model, and to get to use it for predicting new molecule. So this is the step 8. This is the last step where we use this data, where we use this model, which are based on the particular properties and particular molecular data, and we use that to predict new molecular activity. For example, activity, the endpoint, it is also known as endpoint. So like this is the basic principle of anything like machine learning, traditional machine learning or deep learning. So this is one of the good picture which is shared by the data professor. If you go to this website, to this YouTube channel of data professor, you will get many tutorials regarding this data science, how it does, how it cleans, how you can initiate and build the models, and feature selection and everything, how you can predict. So it requires so many steps, like exploratory data analysis, which helps to understand what kind of data it is, what kind of your data. It will give so many beautiful, nice statistical pictures to analyze those data. And one thing here one has to remember that if you input the data, if the data you input for building the model is junk, is a garbage thing, which is not good enough, then you can expect only junk will come out. Means it will predict wrong thing. The accuracy, it will show the higher accuracy, but the data which it will predict must 100% will be wrong. So before doing anything, just ensure that what you are inputting the data, it should be clean enough and it should be a good data. So data splitting and model building. So there are some kind of machine learning things like supervised, unsupervised, reinforcement learning. These are the types of machine learning which you can learn. So these are some of the parts in machine learning. Two types of tasks you can perform, like classification or regression. Classification, where you can classify whether the molecule, for example, the molecule is inactive or inactive. It is a binary classification kind of thing. And regression, where you can find, for example, what is, if it is active, if the molecule is active, for example, then what will be its activity score. So it regression works on the continuous data. So it gives a particular value, it will give a particular score, that how much active it is, what is the score of that molecule. So these are the two things of machine learning which one can do. And there are so many libraries, Python libraries are present to do this machine learning and deep learning, like TensorFlow, PyTorch, Scikit-learn, Keras. So these, some are very popular Python libraries one can use to build their machine learning models or deep learning models. So virtually screen, like I said that in virtual screening, one can also screen the molecules against a particular drug molecule, but one can also repurpose the molecule. Means the molecules, the compounds which are already been discovered as a medicine towards a particular disease, can that molecule will also work as a drug for another disease. So this is also one kind of study we are doing and everyone are doing. So this kind of study is called drug repurposing, where one molecule, like for example, paracetamol, everyone knows it works for fever, but it also works for headache. We can also use it for headache and body pains. So So this process where one drug can work for multiple targets is and how to find that kind of molecule which can be a treatment for various other diseases. This process is called drug repurposing. So these one can do using machine learning very easily. So the AI drug app, which we have, me and my brother, Dr. Vikas Karade, he also helped me. He also mentors me for developing this web app. And later on in September 2021, one more person, Dr. Damini, she has joined and collaborated in our work to build some more modules of AI drug app. So these are some of the features that why AI drug app is so much popular, and how these are some of its applications. Means it reduces the cost. It is very fast because it is based on the artificial intelligence, deep learning. It gives accurate results. It's a kind of lab on lab. Means you can use that this AI drug app from anywhere. It is user friendly, scalable, and large scale problems. And we are still working on it. So this is one of the module of AI drug app where we can predict the bioactivity of the molecule, where the user just only upload their data, molecular data. They just click some buttons. I will show you in this presentation also the actual one, and you will get the predicted result. So we have also included some more modules, some more tools to the AI drug app web applications, like mole identifier, where it converts the molecular names into the smiles, into the structures. And it also generates the descriptors, various kinds of descriptors, the properties which I have already described in previous slides. So using various tools, using various Python libraries like rdkit, moderate, and it generates various kinds of properties in the form of scores and in the form of fingerprints. It also helpful for building machine learning regression or classification models, or deep learning related regression and classification models. So we have also included the bioactivity prediction, where one can predict the binding affinity of the molecule to a particular protein. What will be the score? How the molecule will bind to a new protein, a new molecule. Means we can predict it in the form of scores. So this we have also included. So behind the AI drug app, while working, first we initiated it for building a application related screening of molecules towards COVID disease. So we took those kinds of data which are involved in the coronaviruses protein data. We have collected it from a database known as PubChem, and that data we have used to for building machine learning or deep learning models using various Python libraries like TensorFlow, Keras, which I've already shown. And these programming, this where we connected the dots, is we kept it in GitHub, so that from GitHub we can deploy it into the cloud. So if you will run it in your machine, then only a single person can work on that. So we need to deploy it in cloud where everyone all around the world can use. So this is in a simple term I'm telling you, but there is a very big definition is behind the cloud end, because it's also still expanding and still growing on. So it has also immense value, and there is a different course, different subject of cloud application, cloud platform. So this is just one of the part where we used Heroku cloud application, from where we deployed our web application so that to the cloud, so that everyone can use it around the world. And we have also done some case studies using medicinal plant molecules or antiviral molecules, whether they can be, we can screen these molecules, whether these molecules can act as a drug against the corona or not. And these are some tools we have used to, again, other tools to confirm our molecules, what we screen from the AI drug app. These are some of the publications and awards I got for AI drug app. So this is how AI drug app look like. So I will share once again my screen and I will run a simple. So this is the AI drug app right now I'm showing. Is it visible to everyone? Is this web application is visible to everyone?
Yes, ma'am.
Okay. So this is the first page about AI drug app. Right now I'm showing in my laptop, and you can also check this by yourself in your home computers or your desktops using this link, SARS-CoV app, app.com, if you want to just write it down. So these are many modules are present in AI drug app, like what I said, by activity prediction. It will predict whether the molecule is active or inactive, deep Docker binding affinity, and custom ML tools we have already added. So this is about AI drug app, what it uses and what we have included, and so many tutorials I have also included it here, which I have all, which are in YouTube. I have linked it in here. From these tutorials, you can check how to use these modules for your kind of data sets, how to use this. So all these things are present in here. These are about awards and publication section, and what resources, so what kind of things it does, like by activity prediction for SARS-CoV-19, it what it does. So everything is written in here, and use cases, and when it all these modules were released, all these things we have already captured in here. So these are the people which are involved. So our team is called Tesseractal Gene Team, and this is my brother Dr. Vikas Karade, who helped me so much. And this is Dr. Damini Kule, who later joined us to build the this deep Docker module. She helped me in this. So I'll show you one example. For example, by activity prediction, it will go click on this. So you'll come to this page where you will get more information that what kind of databases, PubChem databases. If you click, you will go to this PubChem database. So this kind of data the model is using. Yes. Yes.
Can you share the screen, ma'am? We are in PPT.
Just a minute. For me, it's showing I'm sharing the screen, actually. Is it visible?
Yes, ma'am. Yes, ma'am.
Okay. Because it's showing me I'm sharing the screen, but I don't know.
Yes, ma'am. You are
Whenever
sharing
you
screen
got
only
this
for
kind
the
of
PPT.
confusion, please. Oh, no, okay. Here it showed me that I'm sharing the screen for the web app also. So this is the web app page. Is it visible? So many
yes,
tabs are open.
Yes, ma'am.
Okay, yeah. Okay, so I went to the bioactivity prediction module. So here you can see how one can use, the steps are written in here, how one can use this bioactivity prediction module. So I'll show you some example. For example, it is taking the input like a single molecule or multiple molecules. For multiple molecules, you can input the molecules in the form of Excel sheet and you can upload it here. But right now I will show you for single molecule. So this is for COVID. We are screening the molecule for against the COVID related protein. So these are the proteins, ACE, replicase proteins, TCR-Pro I have added, and some clinical trials, which kind of molecules can be related, can be similar to the clinical trial molecules which are already in experiments stage, or the molecules which people all around the world are researching whether these molecules can be a drug for the coronavirus or not. And what we want to predict, whether the molecule, our new molecule will be active or inactive, and what will be the IC50, the activity value. IC50 is the activity value. So right now I'll click just activity value. We want to know our new molecule. So the new molecule has, like I said previously, the molecule has some properties like molecular weight, hydrogen bond acceptor atoms, number of, means the chemical structure of that molecule is like that. So these data you can generate or you can get from various other sources, like for example, PubChem. I'll go. Is this visible, PubChem site to everyone?
Yes, ma'am.
Okay. So for example, I'll write aspirin in here. And this is the PubChem database. So it is a database of various collection of various other molecules or drug molecules. So if I'll go to the aspirin, if I'll click. So this is the structure of aspirin. This is the, everything is about aspirin. This is the molecular weight, 180.16. So all kinds of properties you can find from these kinds of databases, or you can generate it from using various tools or softwares like Paddle, descriptors, or you can also generate these descriptors using our custom ML tools. So we have put all these, for example, I have put all these properties. So here it was, for example, molecular weight, 180.16. And after putting, okay, 180.16. So like that, we for our new molecule, for the new set of molecule, which we want to predict whether it is active or inactive towards COVID-19 proteins or not, we have to put all these numbers or scores or properties scores related, and then just click predict. So it will show whether the molecule is active, because we are predicting. In a few seconds, it will show. We are predicting this. What would you like to predict? Whether the molecule is active or inactive. So here it is showing the molecule is active. So right now I just showed, I just input only the molecular weight, but we need some more data, some more molecular properties. So this is the aspirin. Based on the molecular weight, it's showing it is active towards this protein, which is related to the coronavirus, which can affect, which can help to active or inactivate or activate the biochemical reactions for the proteins related to coronavirus. Right now it is showing active, but if we put all those properties, then the result may be different. So here it is showing everything about the models, the train test split, how the data is splitted. Again, the AD information means applicability domain information, where our molecule properties are fit. So it is showing out, means these properties are not coming inside the chemical space, inside the boundary, inside the, you can say the chemical space. Yes, it is. This is the only word I can remember now, because this is a chemoinformatics term. So it's showing whether our molecular with these properties is coming in the chemical space of those molecules which are used for building the model, which are used for building the deep learning model. So right now it is showing out, means the molecule is completely new. It is these kinds of properties are not ever used for building the model. If it shows in, then we can confirm And if it shows in and if it shows active, then we can confirm that the molecule is which we have predicted is confirmed, is a more chances, more probability of being a treatment, of being a drug for that COVID. So we can use these molecules for further experimental for next stages. But right now it is showing out and here it is showing active. So there is a 50-50 chance of this molecule for being a good drug against the coronavirus. And then I'm showing this model summary, how the model working, the deep learning model is working, how many layers it is using. And these are some statistical features. These are some matrix, confusion matrix, or some report which shows that whether the model is good or not. So right now it is showing the classification accuracy of 0.99. 0.99 means 99%. So it shows that the model which we built is and that was for test set, and this is for training set, and this is for test set. So it is showing that the prediction of the model is 94% accurate, means the molecules which it predict whether it is active or inactive is 94%. The guarantee of it is 94%. So like this I have built. So if you want to generate the properties, you have to go to the custom ML tools. You need to open this. Okay, so it's running. And the deep Docker bioactivity prediction I have already shown, and deep Docker is the same as the bioactivity prediction. Just the difference is here you can predict the affinity, not the active or inactive scores, but the affinity of the molecule towards a particular protein. Okay, so it's getting slowed. So till yes, it's running. So custom ML tools have many modules in it, but right now I'll just show how we can generate the properties. So Descal is one of them, where we can generate the descriptors. Descriptors are nothing but the properties of molecule. For example, for single molecule, and which kind of descriptors you want to use, RDKit, moderate, fingerprints, custom descriptors, anything. So for example, if we choose RDKit, and so here we need canonical smiles. So a molecule, the chemical structure of a molecule has can also be shown in a form of canonical smile. For example, in this page of aspirin, where we put aspirin, here we can find this canonical smile. So for here, I'm copying it, pasting the same here, and just calculate. So it has calculated in just few seconds of time all the properties of this aspirin molecule, whatever various kinds of properties it has calculated. You can also download to this. So is this visible now, everyone?
Yes,
Is this
ma'am. Yes.
visible? Is this?
Yes, ma'am.
Yeah. So all these properties you can download and generate for your kind of molecules. So these properties you can again put in here, what I have shown for activity prediction. So this is how. Okay, so there is one Python library known as Streamlit. As you can see in this screen, I have opened the Streamlit website, the streamlit.io. You can go to this link and check this website. This is a very important website. So what it is a Python library which helps to build this web application. It also provides a cloud where you can deploy your web application to the cloud and everyone can use it. So if you want to try, then you have to install this Streamlit Python library to your Python. So first you need to download the Python tool in your laptop, and then you can install this library, Streamlit library, and there you can code it. So Python, this is a kind of Python library that allows to build simple data-driven web application and you can use it for your data science project. So you can just try it. You can check this site, which I have given. And it has also so many examples that how people have used. If you go to the gallery site, then you'll get so many examples here. For example, in science and technology, what people are doing, what kind of web apps people have already developed. So we'll go to this. For example, if I'll show this RA score web application. So this is another web application that someone else has built. So this is okay, welcome. So this is one of the application which I have opened from the Streamlit. And if you want to know what kind of coding is behind this web application, so it will let you into the GitHub, where all the codings were written for building this web application. So R code. So these are the codings one has done in Streamlit library, using Streamlit library. It's just a few lines of codes. So Streamlit is a very famous, very popular because it uses in a few lines of codes. Means you need not to write a long programming. In a few lines of code, you can build so many things. So this is the one of the application you can check from Streamlit website, and there are so many other web applications are also present, and other, if you are interested in other things, so many other subjects, so you can check this site. So you just need to check, or blogs are also, so many people have written so many blogs that how to use Streamlit. See one more cytoscope. They have written some blogs also related to the same application. You can check this out. So many things you can check how to build. So like that, how actually I have initiated. So this is Streamlit. There are so many other Python libraries like Django and Flask are already present, but Streamlit is the best one because it uses very small amount of codes to build the web application. So this you can explore. And in GitHub, this is a kind of GitHub where people shares their codes related to various other projects. So you can just check this out, this page, GitHub page. If you will type in Google, github.com, you will get this. And you just explore it, explore anything. So this is about AI drug app and how I developed this web app. This is a custom ML tool, and others are auto multiml, auto DL. These are for building machine learning models and deep learning models, molecular identifier for identifying the molecules, their smile strings. If you want to, like how, for example, if you want to generate the smile for a particular molecule, so what kind of smile you want, canonical or isomeric? For example, canonical. And if here your molecule is aspirin again, which we found from PubChem database, and get smiles. So in a few seconds, it will give you the smile. This is the smile. So either you can use the AI drug app web application platform for generating the smiles, or you can use other databases. So I tried to compile everything in the same application so that one has no need to go anywhere else to generate the data related to chemicals or smiles. So everything is there in here only, AI drug app. And how I built it, I already showed in using Streamlit and in Python libraries. So all these things, all the information is given here only. So you can check, and publications you can check. Some are free, means I have published in archive. So these are all free. For example, this. You can check these sites. You can check the publications applications. You can read everything in here. One code I have written for automated deep learning. The code you can get from here using this link, and the write-up, everything you can get it here. The tutorials are already available here. You can check these tutorials, how to use these modules, and some more tutorials I will be adding in here. And if you want to know more basics, like what I am saying in speaking in this talk right now, you can check in this tutorial also. This was one of the talk which I have given previously, the same thing. So these are the things. Share the screen.
Question.
Is it okay? Is it visible now?
Yes, ma'am. Yes.
Okay. Okay, please ask questions. So shall I stop sharing my screen?
Yes, ma'am. As your wish.
Okay. Okay. Okay, please. Is there any questions?
Ma'am, what are the algorithms classifiers?
Yeah, sorry.
What are the algorithms and the classifiers of deep learning we have used in our code?
Okay, for AI drug app, I have used two kinds of algorithms for one is classification based algorithms and one is regression based algorithms. These kinds of algorithms where you can related to deep learning, I have used for building the modules of bioactivity prediction and bioactivity prediction. And the algorithms which are used for custom ML tools there are various kinds of algorithms which are related to RDKit, which are related to PubChem library. So these are the algorithms, but for that you need to check that those papers, those publications, which is present in the site, in the AI drug app site, web app. So from there you can get all those informations. So these are the supervised kind of supervised and unsupervised kind of machine learning algorithms I have used. Hello, everyone has understood what I said in the
Yes, ma'am.
in
Yes,
this talk. Okay.
ma'am. You can continue, ma'am.
No, it's over actually. I want to know your questions.
Yes.
Or how to start. So all of you people are from data biotechnology background, biological science background.
Yes, ma'am. Biotechnology and bioinformatics.
Biotechnology and bioinformatics. Okay. So bioinformatics needs more amount of work related to the DNA, RNA, all those genetic kind of work. But this work which I have presented is more of chemoinformatics. So bioinformatics is the one subject, but chemoinformatics comes later, means it is a very new subject. So these all things comes under the chemoinformatics subject, but people are using both bioinformatics and chemoinformatics at a time, and those are called computational biologists. So it has an immense application. And yes, for the same whatever I spoke, the same thing you can apply for bioinformatics projects also, which are related to the data science. Is there any question?
Yes, ma'am. From through which platform you have uploaded to the cloud?
Okay, it's a Heroku application platform through which I have deployed deployed my web application. So you can check the website of Heroku. You will get all the information in there, how to use it, how to install it, everything you can go get, everything you will get. But Heroku, for some amount of data, it will be free, but after some time it will cost you, means it will cost you so much. So if you want to build a small data kind of thing, then you can deploy it through Heroku cloud platform. But if your web application requires containing so much of data and you want to deploy a very big web application, then you need to pay some amount to the Heroku. There are so many other cloud application platforms like Azure. It is also very good, and AWS. These are some non and popular cloud application platforms.
Yes, ma'am.
Any more questions?
Thank you, ma'am. It has been a very informative session, and we are looking forward for having more such sessions, which would be very beneficial for our biotechnology and our bioinformatics students. And we thank you
Okay.
on behalf of all the management and the students. Thank you, ma'am.
Thank you very much for listening to me and for all your enthusiasm. Thank you very much for that. And I would like to connect more. If you need any help, you can mail me or to my email ID, and I'll be very happy to help you. I want everyone should work on AI, using AI thing. Thank you very much.
Sure, ma'am. To be frank, I'm a biotechnology student. I never thought I could be able to learn artificial intelligence app or could build something like that. Now I can proudly say that I'm a machine learning based biotechnologist.
Thank you very much for saying this. You
Thank
made
you.
my day. This sentence made my day. Actually,
Thank you, ma'am.
thank you very much.
Thank-
If I could inspire some people in my background to biotech, because I am also coming from the biotechnology, biological science, and it's a very great honor for me. People can do that by inspiring me. Thank you very much.
Yes, ma'am. Thank you, ma'am.
So, okay, so shall we stop now, or is there anything else?
No, ma'am.
Okay, so shall we end this meeting here?
Yes, ma'am.
Okay.
Okay.
Okay, thank you very much, all your cooperation. So I'm ending this meeting, and thank you very much for listening. Okay.
Thank you, ma'am.
Bye
Thank
then.
you. Bye, ma'am.
Bye to all.
Bye.